I'm really delighted to talk, and Will has asked me to do this for a number of years, and I finally, because I have a mad schedule basically, I've been able to do it this year, and I'm delighted to do so. But I'm kind of also a bit angry coming here because of the future of Will's Institute and the future of the Museum of Leathercraft, and it's just a bit mad. I mean, I've worked, I'm not going to cry. I've worked with both of them, and they're fantastic institutions, and I just can't. I think it's really tragic at the moment, at this time, that we're allowing to let both institutions go. I've worked with both of them. I've learned a lot from both of them. So I'm angry, as well as being delighted to be here. Okay, just let you know that. Okay, so, right, I'm going to talk about stuff. I've got far too much to talk about, so I'm going to talk about a little bit of the stuff, I think. And if it goes on and it's interesting, I'll talk for a bit longer. Why I'm an archaeologist. I study dead animals mainly. People say dead animals are really nice people. So people say dead humans are a bit more competitive. And the thing about dead animals is a lot of them about and the way in which we have transformed our planet in terms of mammalian biomass towards this. I mean, look at that. The wild mammal biomass is really small. And what we have done over the course of approximately 6 millennia has really changed the way that landscape has been managed and to enhance the production of these organisms, for which the hides are generally thought to be what we call a secondary product. And so in zooarchaeology, the study of animal bones, the zoo part of zoos, I guess, what you tend to do is come up with identifiable fragments by comparing these things here with a set, these actually fish jaws, with a set of known standards. So you build a collection and you compare them. And that can be difficult in certain parts of the world where we don't have those reference collections. And also can be difficult as these things get more and more damaged and altered. And so this is just an example of one of my students who's been interested in merino sheep and the evolution of merino sheep as a major producer in medieval Europe. And what she's been trying to do here is look at all of the published archaeological studies on the use of cattle, sheep, and goat. And you'll notice that you don't have sheep here, you have sheep-goat. So sheep-goat ovicaprid exists in archaeology and nowhere else. The reason it exists is because we can't tell sheep from goat. Slight problem. One is a browser, one is a grazer. They're very, very different levels of fat in the skin. They actually make very different products because of different follicle density. Okay, but it's called sheep-goat. And that is 37 archaeological sites with loads of analysis conducted on it. As a zooarchaeologist, these are the kind of questions we're interested in. I don't expect you to take notes. I don't expect you to read them all. But the point is, these are the sorts of questions that we are intrigued to understand this relationship between this small group of fairly unimportant ruminants that become to dominate world global biomass. And if I'm an historian, I would pretty much study the same question, the historian of the livestock economy. In fact, one of the students is a historian, and he worked with the School of Leathercraft, a Museum of Leathercraft. You'll see some of his data. But I mean, basically, so you're kind of talking about the same sort of questions. But the historians do not use bits of animal bone. Okay? They use text that you can just about see behind that. And that was an image of a piece of manuscript from the Norfolk County Record Office. And the Norfolk County Record Office were very kind, and they went through and took loads of photographs for me because I'd asked them a question. Just how many pieces of dead animal, I call them parchment for them, dead animal hide, have you got stored in your collections with a date and location of the legal text written on it? Okay. And it's one county archive in Britain. How many animal skins? They have come up with multiple suggestions. They've calculated and recalculated. But you'll notice these boxes just full of legal documents, because the way that the Brits work, we love writing everything down. We love paying lawyers lots of money. And the lawyers, because they charge us by the word, produce very long documents, right? You know all of that. So their estimate in one county archive is 12 million dead hides. 12 million hides. Do you know how many hides? This is mainly sheepskin, I have to say. I won't talk about that now because Richard Fitzneal, in the beginning, the late part of the 11th century, said we must always write legal documents on sheepskin because it brooks no erasure. And that's actually because you would understand this. You know what? You know sheep hide. You know the fact that there's secondary follicles and the extra layer of sebaceous glands means that you can delaminate sheepskin. And that's why we think that's why they've insisted on using sheepskin in Britain since that period of time. It's almost exclusively sheepskin. And extraordinarily, we've been analyzing a monastery, one of the foundational monasteries in France, and they switched from cattle to a calf to sheep at almost exactly the same time that Richard Fitzneal is writing the Dialogues of the Exchequer. By the way, he writes that because this is the great war between the great sort of war with William and Maud. It's not Maud and I can't remember. And they both have destroyed the Exchequer, so they had to come back and re-establish it. So yeah, so there is 12 million in one county archive, and there's 13 million killed on average, there's thought, each year during the medieval period. So that's kind of like one full year's worth of every single sheepskin gone to parchment, which is kind of like a crazy number. And so just very quickly, you've probably heard of two terms. This is untanned, remember, parchment and vellum. And being untanned, it has to be kept much more carefully because if it gets wet, it'll start to rot because it's not untanned. So what about parchment and vellum? So I just want to explain this briefly. So this is a question that has had a very long-standing debate because some of this stuff is so beautiful and so fine. This is the most expensive computer that you would think a student would ever use of the modern day. This is a tiny, tiny object called a Paris Bible or a pocket Bible. They were produced in the 14th century and the late 13th century. And then when the Black Death hit, there were so many made in Paris, Oxford, and in Italy that they didn't make any more. They just used them again and again and again. But this is the whole of the Bible, Old Testament to New Testament, which is able to be carried by a student, a Pandect Bible. And so what we find here in this analysis of these pocket Bibles is that it was always thought they were done on uterine vellum, so dead young calves. But what you'll see is actually in Britain, it's a mix of calf and goat. In France, it's mainly calf. In Italy, it's mainly goat. So in other words, they're using the local resources. It's just actually about craft skill. It's not really about the actual nature of the material. So it's not all vellum, which is supposed to be calf skin. It's actually just whatever the craftsperson has got. And we actually look at, and so this was done using this technology called Zooms, which now has its own Wikipedia page. I'm glad to see, and there's a picture of a student conducting the analysis. Why do I show that? Well, because it's one of these things where another researcher in 2002 developed the methodology, basically, but he patented it. And so he made a technique which was designed to look at feathers and fur. And the reality is, there's not that many people want to pay to get identifications of feathers and fur. There's a little bit of wildlife. So that didn't do anything. So what we did is we kind of open sourced this and we produced it. And it just went gangbusters, basically. And now this is a really ugly looking slide, I hope to agree. But this is a brochure from a commercial company now, which is actually selling the technology in the States. And we actually have a price because of that. So I'm going to explain the technology. The technology is dead simple. You basically take peptides, you put them on a mass spec, and then it's a laser that gets fired. And then a time-of-flight tube basically tracks the movement of peptides from your collagen sample. So you have taken a collagen, extracted it by gelatinization typically. You then cut it with an enzyme trypsin into small pieces. And you end up with this, if it works. No, I have to go and click that one now. Into the next slide. Yeah, and then you get this spectrum, which is here is the mass over here. And then here is the intensity. And each of these peaks is then used to identify the species. In this case, that's a cow collagen, so cow hide, cow parchment. And the way it actually has worked is, you all know collagen better than I know collagen. Highly repetitive structural molecule. And because of the way that it has that amazing quarter stagger arrangement, you always have these arginine and lysine residues in the same place. Okay, except in some fish, there's some fish down there. And what that means is trypsin cuts at these charged residues, and those charged residues are in the same place because you need the quarter stagger, it's a charged interaction. And it means that whatever species you've got, they'll always give basically the same pattern because collagen is highly conserved. But in the odd place, for instance, you've got an A and a P there, you've got an alanine and proline there, that will change the mass locally of that peptide, and it'll shift one of these peaks. And so it's a really, really simple, fast technique to do the analysis of species. And now we know the price. This is that American company, that horrible brochure, but they will charge you $52 in the States to do a species ID this way. And this is just a plot. This is also, a lot of people put stuff on Wikipedia. This is a Wikimedia Commons illustration. And the talk I was mentioning on the Parchment was this one, so Finament, who was able to analyze many, many Parchment documents without cutting them. And the reason you don't cut them is it was actually a desperation measure. We talked to the conservators. We had thought we'd be able to go into that massive archive. They got 12 million documents. They could easily give us a few. No way. The conservators would not let us sample anything. And so Sarah, in desperation, analyzed essentially the waste buckets around the conservation chili. So all the waste products of conservation. And one thing they do is use nothing more exciting than the humble Staedtler Mars eraser, that white soft eraser, to remove dirt from the surface. It works very well. And so they would have these little piles of razor waste in the studio. And we discovered this traps proteins and DNA really well. And so we could just ask the conservators, please, could you just conserve the waste, don't throw it away, put it into tiny tubes and send it to our labs. We've been doing that now for about 10 years. We had 7,000 parchment samples analyzed, courtesy of the conservators, not us, right? Anyway, so that was that technique. And so that was the study. And what we were able to show was that we have this very mixed production. It's about the skill of the parchment maker. It's not about the animal. And this just shows you the kind of skill. These are in millimeters, the thickness of the individual folia. They are they're like tissue paper fine production. And you'll notice that with the cast skin, the blue, you produce more of these very, very fine skin. So that it's true that the vellum produces the finest quality parchment, but you can see that you can produce it from all of these different animals. So Zooms is so simple that school kids do it now. So in Denmark, where I also work, there are large foundations like Novo Nordisk. The great thing about Denmark is all these big companies are actually charitable foundations, 51% owned by charities, and they have to put a lot of money back into research. Novo Nordisk, you may know, that I think the richest pharmaceutical company in the world. Their GDP is greater than the whole of Denmark. And so their profits go into the whole of Denmark. Anyway, so they've been putting money into this thing called Next Generation Lab. And what this has been is because when they put the metro in Copenhagen, they found a lot of leather. You probably know, I'm sure you all know, from the work of people like Ray Thompson and others, that leather preserves extraordinarily well. Thanks to the skills of you, the great tanning skills, leather preserves in anoxic conditions so well that the archaeologists just got bored and they couldn't cope with the quantities of leather they were producing. So they're going to check it out. So my colleague Lisa Oster-Blanc said, well, we'll analyze it for you with school kids. They said, great. So now what happens is we have now 2 freezers or fridges full of leather from Copenhagen, from different levels, from different dates. And the school children go along and they analyze the material. They do a visual analysis. We have leathers prepared, so they do first a visual analysis, and then they will come and they will do the zooms analysis. And we get 2 different schools to do the same analysis to check, quality control. And they've already found here, and this is a discovery that you can never break the law a bit. So in this case, they're not supposed to use horse to make leather in Copenhagen in this period. But what do we find? We find horse leather. So it cheats. But this was done by, the authors are shown here. But I put Skulkins in, because actually the Skulkins did all the work. And they've used it with local museums now. So the museums put exhibitions on about this stuff. So, it's really great. Zooms is so easy. As I say, even Skulkins can do it, And they get the difference between thinking about, the visual appearance and then thinking about the chemistry and then thinking about the time and the role that leather had to play in the Copenhagen economy at the time. So it's a really nice study. And Novo Nordisk have just given us more money, or at least more money, to a second tranche of this. They've built a beautiful laboratory. I should have shown a picture of that in the museum. The back half of it's got sort of Bruegel paintings. It's got old leather. It's got a lot of working tools. Then the other half, you've got modern day robots doing all the spotting chemistry and all the lab stuff. So the school pupils, a fantastic experience. Anyway, so what I want to think about now, we've got some technology we can play around, we can do stuff. This is Saxel's 1954 master's thesis from the University of Leeds. I should have credited her. I haven't credited her here. And she was a mature student at the time. And she went to visit Russell and Hitchin. Russell in Hitchin and Hart. She went to visit the Parchment there. And when we talk about the fact that there are 12 million estimated sheepskins in Norfolk County Record Office, what you've got here is the fact that we've got the production and the sorting. And what happens here, you can see this, the best linings, stretched out and split side, are selected to make parchment. Others are then made for chamois leather or into doeskin. So in other words, that 12 million represents the best quality parchment, presumably from the light leather industry at the time. So the light leather industry is preparing these skins, and they're selecting the very finest ones with the fewest blemishes, because these are very large legal documents. Each is a single hide, and at least the more recent ones are. And of course, because you're paying by the word, there's lots of words on them. So we think about this process for production. We can ask questions about every single aspect of that production, and then we can start to look at what kind of evidence we can draw from the hides themselves as to the story of production. And so, if we go back to this first question, which was when and where were different animals being used, this is what we got with the animal bones. We had all of this data, and we didn't get very much taxonomic resolution, and we didn't get very good temporal resolution, because the bones can't tell you what the animals are, sheep and goat, and also we don't know the dates. Of course, if we look at the parchment, we get a result like this. Very high chronological resolution to the year for legal document. We can tell sheep and goat because we're using zooms. And so one student in a small part of her project can do a much, much more comprehensive analysis, at least for the production of parchment. And somewhat unsurprisingly, given the importance of wool in the Spanish economy, the vast majority of documents are written on sheepskin. This is although we, and anyone that works as a scribe will tell you, goatskin and calfskin are better and command higher prices. So this is about accessibility of product. And so what you see in England, which is again also massively dominated by sheepskin, but again, we still do not know how this single son of the Bishop of Ely, Richard Fitzneal, who asked that all the English documents are written on sheepskin, impacts across the rest of Europe. We've seen it now in France, but what we do know is the one place we see legal documents not written on sheepskin is, of course, Scotland. They use goat and calf. So I don't know. So no, that this impact from the English which impacts the continent, we don't know how far that goes, and whether these legal documents are written on sheepskin for the same reason or for other reasons. The other thing is, we can see evidence for butchery, and we can see evidence of flaying. And this is an interesting example of my colleague Annelise Genois from Sorbonne. And this is a car. There's a cow skeleton here. If you look, you can just like see here's the ribs, the legs here, and there's a calf which has died during the birthing process. So it's still in the birth canal, and that kind of gives you a better image of what it was. And what's interesting about this, you can see that the cow was flayed for its skin, but they didn't recover the calf for its so-called really high quality fetal calf skin vellum. So in other words, this also tells us that our view that somehow, this unborn calf skin is the most valuable thing is probably not true, at least by this time, which is 14th century. I'll skip through that one. And what we discover from Annelise's work, looking at English legal documents primarily, is there is a passion for digging up animals to get their skins. So she has a whole series of legal texts like this, which go on and on about, look, this animal has died of some horrible, nasty plague. We've buried it in the ground for a reason. Stop digging them up and getting the hide to make into leather. And so they start doing things like they start quartering the animal, they start putting cuts in the hide, because people are really, the demand for the hide for leather production is extraordinarily high. And so you can see with the cow that died, they took off the hide of the cow. They weren't interested in the calf skin for the parchment. So if we need to actually understand that more, we have to go beyond simple mass fingerprinting. We have to use this, that was just mass fingerprinting. Here, we're going to go this thing here, which is they've got two mass analyzers. What this does is it takes those peptides we analyzed previously, and we're now going to do more detailed analysis. This more detailed analysis means we're going to introduce them using a liquid chromatographic system. We're going to select out an ion of interest from a mixture here, throw most away, keep this one, or that one rather, keep that one, and we're going to take this one and we're going to smash it up. We're going to fly it through a cloud of gas, poor little thing, kind of inert gas, and it'll break into fragments. And when it breaks into fragments, what we can then do is we can analyze those fragments, and we'll get a series of masses from the fragments, and rather as before, but these are fragments of the peptide. These are not fragments of the whole protein. So what these can do is actually tell you the amino acid sequence. You can actually now read the sequence of that peptide. Okay, so this is a more expensive, more involved analysis, which will cost seriously more money to conduct the analysis. But we use that because what we need to be able to do is ask the question: can we see from the vellum evidence of the age of the animal? Because we can look, and we can actually get changes in the circulation of proteins in the blood, from the fetus to the animal. And what we're able to do with that is verify that the calf skins we're looking at are of very young animals. And in fact, we know by measuring the size of the animals more than actually from the fetal markers that we get both, that these animals are killed to make vellum at between 6 and 8 weeks of age, right across. So all of the calf skin we're looking at, the sheepskin is adult animals, but the calf skin is really, really young animals, which is kind of very curious because you needed in those days the calf by the mother for the so-called let-down response to allow the cow to milk. And you're slaughtering an animal when it has almost no economic values. It's a very young animal. They grow very rapidly in the first 18 months. Even in the first 6 months, there'll be much more meat. So you'll get the rennet out of the stomach, which could be used for cheese making. You get the parchment, but we're kind of surprised by how many of these animals are killing for parchment. And there's a whole story about that, which is just about to unfold. I won't talk more about that in a moment. I'm going to take a slight turn around because it's just a cool story that came out last year. And it uses, again, LC-MS. It's just to highlight the requirements of sensitivity. You've heard of a guy called Herodotus, I hope. He lived in the fifth millennium BC before Common Era, and he is known as the father of history and the father of lies. He lived here in modern-day Bodrum, and he then this is the world as he describes it. This is the world. I mean, he traveled very widely, and he's highly criticized often. Up here in modern-day Ukraine were the Scythians, and he writes quite a lot about the Scythians. He seems to know the Scythians well. And they're up there. If you take, here's the map of a series of these Kurgan burial sites. And what he says about them is they're pretty gruesome people. And he says how when they murder their victims, they drink their blood, they cut off their heads, they skin them. And one of the interesting things he says about them is they take off their skin, nails, and all, and from their enemy's right hand, and they make coverings for their quivers of the human skin, because it's thick, bright, and it's whitest of skin. And it's true that if you go and look at the quivers in these Kurgan burials, and there's a picture of the quiver here, and these are examples of the quivers, they are very light-colored leather with very bright reds and blues on them. So they're very kind of starkly illustrated. So anyway, well, it's leather. It's could be human skin. Herodotus, is he right or wrong? And the answer is he's right, but only occasionally. So this is the analysis of all the leather from those quivers. And you'll notice that 4% is human skin. So Herodotus, father of lies, he's right in this case. And the way we could do that is we can do it with zooms. It's actually pretty clear. Zoom spectra, those markers are markers. But to make sure, because we couldn't quite believe it, we then did full LC-MS analysis, which is the more involved one, just to make sure we had all the right peptides to say this is definitely human. But so that was a kind of curious one. So okay, now we've got the tools, we've got the zooms, we've got LC-MS, we've got an awful lot of parchment. What can we begin to learn about the parchment? And so this is, of course, where we've been interested in the process of craft, really, how these things are actually produced. And so one of the things that we can do is, and the moment we can only do this with cut pieces, which I got some very generous people gave me cut pieces of parchment. These are all cut, dated legal documents. You can look at the health of the animals. And this is something I'm trying to get funded further, so far failing, by using a new kind of technology which allows me to do this incredibly sensitively just from the eraser rubbing. What you're seeing here is two isotopes, carbon and nitrogen. Nitrogen is telling you about essentially how much the animals we know were stocked on the land, and also a little bit about health. And you can see some nitrogen values here, which is extremely high. This indicates probably unhealthy animals, which is what I'm intrigued about. And you can see here the nitrogen offset. We've got some very elevated values in animals of poor health, not one that dies. So there's a stress marker there. The carbon, not so interesting, except this is kind of curious. And this speaks to the demand for leather. You see a change in the carbon isotopes here. And the only explanation we can come up for this is the fact that some of these animals are not based in Britain, or they're being fed on crops that were not raised in Britain. And using so my student historian working at Museum of Lovecraft, he has come up with the fact that what we're seeing here is in the period of the Napoleonic Wars, a massive increase in demand for leather. And what we're starting to see is more and more animals coming in from places like Uruguay, where they're feeding on different foodstuffs. We think what you're looking at here is these elevated values, still got low values coming on here, but these higher values are animals which are coming from probably South America, just to feed the British leather industry. Okay, so that's something one can look at. Probably doesn't interest any of you that much, but it's kind of intriguing. And the stress one is potentially very interesting for us. So that's talking to us about the selection of animals, what animals we're choosing. We're choosing the local animals. We're choosing the sheep or the goat or the calves. And we're having to import stuff for production at times. The second thing is, how do we dehair the hides? So you know that the most common way to dehair, at least in the past, now the additional tools we use, is use lime. And this is just some data on the duration of liming. And that's a modern day skin, 5 days, 10 days, and 15 days in liming. So we do a lot of We try and make parchment, particularly Erzu's Nucleus is the star of this. And we try and make it using all these different kind of medieval recipes. We're trying to understand what the mechanisms of production are. So what we're seeing in the liming is one of the things we're picking up here is this amino acid, glutamine, deamidating to that amino acid, glutamic acid. So it changes the charge. That also changes the mass, and changing the mass makes it very easy for us to measure. So it's something that automatically comes out of analysis for free. And we call it the PQI, the Parchment Glutamine, Q is glutamine index, because they're dealing with a lot more of these mulesta type animals with very fatty skins. And if you have a very fatty skin, you need to saponify them for longer to get rid of the fat. So maybe that's it. We don't know. But the point is, you can see changes in production on the longue durée. Oh, sorry, yeah. And so then what this is looking at one single library. This is a library of the monastery. And you can see that this is the PQI index. The calf, much less time in line. Goats, the longest time in line, I guess, and sheep more variable. So the liming times show differences in the different species. And I'm kind of surprised about this because I would have thought, again, based on the fatness in the skin, that we'd see more of the sheep skin with the lower values than the goat skin. And intriguingly, when you analyze the DNA, you get almost exactly the same pattern for DNA damage. So the DNA damage is mapping onto the collagen data. And another thing about that, this is samples supplied by Deborah. What we've been doing is we can use this to very quickly analyze the extent of deamidation of individual sites on the collagen chain. So this can then give it, and what we've been looking at, and one thing's about the parchment called the index, it looks at the relative rates of deamidation on individual positions within the collagen, and then it produces an aggregate value. So it's incredibly powerful. It's a very, very simple technique of just fingerprinting the sample will give you very great detail about the quality of your product during processing. The second thing about it is we don't just analyze the proteins, we also analyze the DNA. Both come up in this eraser rubbing. And of course, we're analyzing the surface of the parchment. So we're kind of analyzing this. We're analyzing the microbial forest, which is living on the surface of the parchment. And what we're therefore looking at primarily is the mechanism of parchment production. And what we are seeing, if we look at Southern European parchment, this is my colleague, is we are looking at salt-loving bacteria. So these are extreme halophiles. And we don't know why these extreme halophiles are on the surface of parchment, but of course, what we assume is this is coming from the salting of the skins, right? So we're looking at salted skins. And if we start to, and the intriguing thing about this is that in the past, this is the logarithmic scale of salt prices in Sweden. As you go further north, salt prices are getting higher and higher in the medieval world. Salt is much less accessible. This is a picture of a series of salted skins from a parchmenier. And so what you can then do is you can analyze all the bacteria living on the parchment. And what you can see here, these ones which are in the green here are ones which are salt-loving organisms. And you can look at the pattern of these in different parchment samples. And what we notice here is this is what's called a principal components analysis. We're trying to separate out a lot of genomic data. Actually, one of the things I should also say is that the microbial diversity is very low. It's kind of limited on the surface of these parchment documents. You've got here, principle 1 and 2 account for massive amounts of total variance. And we're separating this out between traditional parchment making, shown in blue, and Icelandic parchment, shown in purple. So that is just Iceland. And what's special about Iceland? There's a joke about Iceland. What do you do when you get lost in a forest in Iceland? You stand up. Basically, the Vikings destroyed almost all the forests on Iceland. What does that mean? It means although you've got vast amounts of seashell, you can't turn it into lime because you haven't got the fuel. So that without any lime, they produce parchment in a very different way. They rot the skin down. And what you're seeing here is the microbiome of a rotted or retted skin. Okay? That's why Iceland is so very different. If you then look across the rest of the data, this is English material from medieval, and then this is also English, and this is all Spanish. And when we look in more detail at that, what we're looking at is differences in production. So we've got local production around Yorkshire here, and then we've got a range of the products here. And what we're seeing in these ones, the reason these are causing big separations, is the diversity of the salt-loving bacteria. And I don't know, this is very early days. We want to do more on this. I think it's actually tracking where the salt is coming from. It's because in medieval and later Europe, mined salt from a cave is known as white gold. It's incredibly expensive. It's very pure. It's used for culinary practices. Dirty salt is the salt which is coming from salt pans, and that's the stuff that should be used in these skins. And I think what we're tracking is different sources of salt, I think. What's my time like? Okay. So, go on. Whoa, okay, fine. I can keep going on if you want me to. Right. So the other interesting thing that I think is really intriguing about this is the presence of this organism, which is, we found this, a complete genome of this organism, one of our pieces of parchment from Hull. This is from 17th, 18th century parchment. And I'm slightly worried about it because it's almost identical to this particular strain found in pigskin. So maybe it's a modern contaminant. We need to do more work. A colleague of mine has found a very, very similar organism in the teeth of sheep. And it's known to be a problem for people working in abattoirs. It's a common pathogen. And so if you think about, and this is why I need to talk to you more than myself, if you think about the way that you prepare hides, you have got lime pits, and you have to think back into your fathers and forefathers. You have lime pits, and honestly, how often do you really clean out those lime pits? Okay. And when we read about medieval lime, we have grey lime, we have quick lime. So basically, you have the lime pit, which has been overused, so it's for saponification primarily, so it hasn't got much bite anymore. And the skins are put in there to basically clean them off and degrease them, and then it goes into quick lime. And what I imagine is happening here, and I may be wrong, so I'm talking to you, is that we've got in the abattoir, in that grey lime, we have a whole microbiome community emerging. And all these animals, while they're salt tolerant and they're alkali tolerant, so they must be organisms which are coping with the lime pit. And I think what happens is you take that skin out of that grey lime and you put it into the quick lime, and the strong alkali in the quick lime is basically hydrolyzing all the enzymes, hydrolyzing all of the DNA. So when you take the skin out and dry it, you've taken the DNA and you've entrapped it in the skin. This DNA is almost undamaged. So I think we are sampling essentially the lime pits of the parchmentaries. And if that's the case, that would be utterly remarkable, because we could then look at essentially the history of individual sites of parchment production, fingerprinted by the lime pits they have. And of course, those animals that are getting selected are the halophiles from the salted skins, which are also alkaline tolerant, because they're living, I think, in the lime pit. I think that's the story, and I'd love to know more about it. But this is a discovery we've made very recently at the end of our funded project, so I don't know where it is, where we can take it, because we've got no funding for it. But it kind of intrigues me that we are looking at potentially the pathogens associated with abattoirs. At the moment, if you go to America, there's a very large funded project looking at the pathogens in abattoirs. They're interested in looking at things like antibiotic resistance. And it's kind of extraordinary to me that it may be the case that these parchment samples, the 12 million Norfolk County Record Office, are essentially dated records of the pathogens in abattoirs over the last 800 years. I throw that out as an interesting observation. So, and the very last part, I've got 10 minutes. Okay, very last part is, so that's all fun. That's really interesting. And we are beginning to explore the nature of this incredibly rich and overseen record of dead animal hides. But what are we missing? And what is it interesting, and this is what kind of my obsession at the moment, is if we look at proteomics experiments, these are two very large studies where they analyze all of the identified peptides from that LC-MS and MES experiment, that second video you saw, when you take the peptides and you fragment them. This is 25% identification. So it meant when they did that fragmentation, they showed at the very end a very nice sequence with some fragments. That only happens 1 in every 4 times. 3/4 of the time, they can't identify what that thing is. Okay. If you go to ancient samples, and these are various ancient samples, you find it's much worse. So we are really bad at identifying things. And the interesting thing there is, if you're preparing leather, parchment, you're actually modifying the skins. That's what you're doing. You're modifying the skins to make them resistant to biodeterioration, to change their physical properties. And those modifications are leading to mass changes in the sequence. But at the moment, there are no software algorithms that allow us to see what those are. And we see that because when you look at these ancient samples, these are modified, not in the same way, these are modified by damage. But the reason we're not able to see those peptides is because they've been modified, they've been changed. So my focus has been on really trying to understand what are these software tools doing and how can we do them better. So this is a really dumb experiment we did, which was a very simple protein, and we just smashed it apart, and then we analyze it using different software tools. It's a complicated graph. We have fresh samples. It's actually fresh ones after 4 days and 128 days at 95 degrees centigrade. So we're really heating these things up and smashing them up. And what you're looking at is the higher the line is up here, this is a fraction identified, none here, 60% here. So the higher up the line you go, the better. And what this is a line where you say, it's called a false discovery rate. It's if you give it gobbledygook, which you know is the wrong answer, how often does the software mistakenly think you found something? So basically, these are different false discovery rates at 10 to minus 2, 10 to minus 2, 10 to minus 2. I don't know. Anyway, what you'll see is that most of the software tools, and these are the two that mainly use, they're really bad, very, very low lines. And then some new techniques which have got much, much higher signals. So we're focusing on the ones with the higher signals. But even then, the very best ones are only about 25, 30% identification, which is accurate, even when we know the right answer. So no one else seems to have done this. They put in a very simple protein, and they tested with all the software. They discovered that none of them very good. I won't go into detail about the other various, but these are all different ways you can search the same data and get different results just by changing some of the parameters in the software. So it's not very good. And so what we've been looking at is transformer models, and I'm sure you're all familiar with these now. And what these are is they're based on the concept of attention, which is basically positional dependency. So they're neural networks which work on, that's my 45 minutes gone, on dependency. And what they attempt to do is use the information from the last word, the last token, to predict the next one. And this has been widely used in language models for language, but amino acids are a kind of language because these proteins have got very similar order, very similar structure, very similar patterns. And what we've been discovering is we've started, these were only released, the first one in December of 2022. We tried it in February 23. We're blown away by it. And we've basically been focusing all our attention towards how we can begin to integrate this kind of technology into our understanding of protein damage. And so what I failed miserably to do to you, that this just shows you that they're getting better and better and better. That this is the accuracy of the peptide level and accuracy at the individual amino acid level of a whole series of these things all produced in the last 18 months. So a massive expansion of these models. But what's interesting to me is this. They're still not very good at what they call, no, they don't call it PDM, but these things these are normal amino acids, and these are modified amino acids. So this is the deamidation we talked about, the thing that's associated with liming. they're really, really bad at identifying such a simple chemistry as that modification. So what we're now doing is writing software to basically address this issue so that we can deliberately look for patterns of damage. Why? Because for us, it's really important because all of our ancient samples are damaged. But I think it's going to be important for people that work on modifying proteins, as you do, to change their properties, because the current software is just not fit for your purpose or ours. So that's what we're mainly focusing on now. And I'll leave it at that. But basically, we call the thing valley indexing. Our view is that everyone else looks high from above on this planet of peptides, and what we do is dive deep into the valley to look at all the stuff and modification. But that's and I'd hoped by today I've had some really cool data on some stuff that that's been shared with me from people here on their analysis, but I haven't finished the analysis yet because the software always takes me longer than I think to write. So anyway, I'll leave it there, but that's where we're hoping to go to mine more from these hides, to learn more about modifications, and then apply them to both modern and ancient skins. Okay.